ORFfinder. ORFfinder will take your DNA or RNA sequence, nucleotide sequence, and it will search it for all the ways it could be used to make protein. And it's got different thresholds that you can set to only look for big and probably real proteins, or to look for even fairly small proteins, stuff like that. All right, so bam, all you do is you paste in your sequence. If you only want to search part of your sequence, you can limit it here, but you usually don't need to do that. That would be if you have like a genome of millions of bases and you only want to look at some little part of it. This is the important one right here. So you want to set the minimum open reading frame length. So if you set that at 30, that's 30 nucleotides, which is 10 amino acids. And that is a real small protein. That's about as small as you will ever see. For a protein coding gene. A more normal size is going to be something like 300. That would be appropriate for both prokaryotes and a little on the small side for eukaryotes, but you probably wouldn't miss much that way. And so, yeah, that's usually a reasonable thing, but we're going to miss any open reading frames of less than 300 nucleotides if we do this. Okay, the other thing to look at, you're not going to mess with the genetic code because we're going to be working with things that use the standard genetic code, which is most of life. There are only slight tweaks out there. A lot of them in either bacteria or mitochondria, which are bacterially derived. Okay, open reading frame start codon. Seems like a no-brainer, right? You say, well, yeah, my open reading frame starts with an ATG. Question is, do you have a complete open reading frame here, or do you have a chunk from the middle of an open reading frame? And if the answer is, well, I don't know, then I would say you go on here to any sense codon. If there is an open reading frame, whether or not it has an actual start, because the start may be before the sequence that you have assembled or found, then this will actually still pick it up, which is useful. Ignore nested open reading frames means that if you have a larger open reading frame, it cannot have smaller open reading frames lined up inside of it. I generally would not tick that one. So this is a pretty good sort of standard search. Let's submit it. There we are. We found 5 open reading frames. Fantastic. And so look at that. Look at the size of that one. It's enormous. So which one is that? Why, that's going to be the first open reading frame. It says it is 8,070 nucleotides. Seems unlikely to occur by chance. This will give you the protein. So you can click on open reading frame 1 and you can just copy that protein. Bam, right there. Look, they even put it in FASTA format with the little title that starts with a greater than. How cool is that? You can also pull up all these other things, which probably aren't real, but darn it, they are in the sequence, and so they are potential reading frames. And so you can copy any one of those. You can download the whole set of these things. So here, mark subsets. Let's mark the longest 3. Great. And you can download all of those at the same time, and it would give you a nice little FASTA formatted protein database. You can take these right now and you can blast them, or you can do smart blast, which will just blast the top hits for your thing anyway. And so it's kind of a little one-stop shop, but it comes from the same people that run the blast servers essentially. And so that's how it's able to tie into those so nicely. And it's just using the Swiss Prot right now. And you could maybe switch it over to NR. It doesn't matter. They both have a ton of overlap. Pretty much everything in the one will be found in the other. So it doesn't really matter. And there you go. That is ORFfinder, the open reading frame finding tool.